we are going to do is that we are going to solve the topical MCQs of the chapter of immunity, which is from the AS Biology 9700. Let's look at question number 1. It says that which row is correct for neutrophils and the B lymphocytes. In order to solve this question, we must first have the knowledge about the white blood cells. Let's now move on to the lecture slide for the white blood cell. We know that white blood cells are also known as the leukocytes. White blood cells की अगर हम classification की बात करें, so white blood cells are further classified into phagocytes and lymphocytes. And if we talk about the further classification of the phagocytes, we know that there are two types of phagocytes, white blood cells, that are the neutrophils, and the macrophages, which are also known as the monocytes. And then if I talk about the lymphocytes over here, we know that lymphocytes are classified into B lymphocytes as well as the T lymphocytes. And then the T lymphocytes over here are further subclassified into T helper cells and the T killer cells. What is important for us to realize that we must know the functions of each type of the white blood cell we have written over here. Let's now start with discussing the neutrophils first. Acha, we know that neutrophils is having a lobe nucleus over here, as we can see, and it is also having a granular cytoplasm. Why it is having a granular cytoplasm, or how it is having a granular cytoplasm, we can see these small dots over here, which is representing a granular cytoplasm. It do not have a clear cytoplasm. The next point which we must know about neutrophils are that they are the short-lived cells. They are not the long-lived cells. They are the short-lived cells, and they can reach the site of the infection by squeezing through the capillaries, which means that they are having the ability to change their shape. That's why they are able to reach the site of the infection. Then, if we talk about the term, if we talk about the type of the phagocyte, that is macrophages या फिर monocyte, we know that they are having a bean-shaped या फिर kidney-shaped nucleus over here, and they are also known as the professional antigen-presenting cells over here. They carry out the phagocytosis, which is basically the process of engulfing the bacteria या फिर the unwanted substances. अच्छा जी, lymphocytes की if I talk about the lymphocytes, so lymphocytes nucleus covers about 90% of the cell space. As you can see over here, this is the nucleus and it is covering the 90% of the cell space. It has the peripheral cytoplasm, which means that the cytoplasm is squeezed at the edges of the cell. Now, if I talk about the classification of T lymphocytes, which were the B lymphocytes and the T lymphocytes, it is very important and very high yield for the MCQs to realize that the B lymphocytes are the cells which make the antibodies. We are going to discuss it further as well, that B lymphocytes, when they identify any antigen, what do the B lymphocytes do is that they further divide into the plasma cells as well as the memory cells, and then those plasma cells are the one that release the antibodies. So in short, we can say that the B lymphocyte makes the antibodies, and this is a very high yield question. Yeah, it is a very high yield statement, you can say, for your MCQs. Then if I talk about the T lymphocytes over here, T lymphocytes there comes the T helper cells and the T killer cells. If I talk about the T helper cells, what do the T helper cells do is that they release the chemical which is known as the cytokine. Because cytokine is that chemical which activates the other cells of the immune system to carry out an immune response. It helps to activate the immune system basically. The helper cells are also known as the king of the immune system because they are the cells that are activating the other white blood cells of the body in order to carry out an immune response. If I talk about the T killer cell over here, what do the T killer cell over here do is that they destroy the infected body cell, for example, the viral infected cells. Okay, those cells of the body that are infected by the virus or that are infected by any other pathogen are killed by the T killer lymphocytes. Now, if we move back to the MCQ page, we can now more easily answer the MCQs. Which row is correct for the neutrophils and the B lymphocytes? Neutrophils ka we have just discussed that it can change the shape, and B lymphocytes are activated by the contact with the antigen. Obviously, B lymphocytes are having the function to produce the antibody against a specific pathogen, and when it will produce the antibody against a specific pathogen, when the B lymphocytes will come in contact with the antigen of the pathogen. So, we can over here say that option A is our correct answer. Let's move on to question number 2. It says that when exposed to an antigen for a second time, memory cells stimulate a secondary immune response, which correctly shows the secondary immune response. Acha, we know a thing about secondary immune response that it is more rapid, right? And what happens in the secondary immune response? In the secondary immune response, because there are the memory cells against a specific antigen stored in our body, what will happen that the immune response will be more rapid, and there will be the more rapid division and the differentiation of the antibodies than the primary immune response. So we know that if there are the antibodies being secreted, so we know that this is the memory cell for a B lymphocyte. And we know that during the primary immune response, the B lymphocyte splits into the plasma cells as well as the memory cells. The plasma cells secretes the antibodies, and the memory cells are stored in the blood for the future. In case that the same antigen attacks in the future, so the memory cells can carry out a secondary immune response, like they are telling to us in this question. When the memory cells carry out the secondary immune response, what do the memory cells do? Is that they are again further going to divide into the plasma cells and again the memory cells. And these plasma cells are then again going to secrete what? Antibodies. Also, do remember that the memory cell itself is not going to secrete the antibody. Memory cell is going to divide into the plasma cell, and these plasma cells are going to secrete the antibodies. We cannot over here say that the memory cell is producing the antibody. This will be wrong. So हमारे पास option A will be the incorrect answer. Divides into the plasma cells. It is dividing into the plasma cell, but it is also dividing into the memory cell. Okay, so we can say that it divides into the plasma cells and the memory cells. Hence, our answer for this MCQ will be option C. Let's have a look at why option D is also our incorrect option. Secretes the antibodies and divides into the memory cells. No, we know that the memory cell itself do not secrete the antibody, but what happens that the memory cells differentiate into the plasma cells, and then those plasma cells secrete the antibodies over here. So yeah, I hope that this is clear to all of you. Alright, now let's move on to question number 3 over here, and let's see that what they are asking us to do in this question. A blood cell count can indicate how many white blood cells are there in the blood. Where else in the body are the white blood cells found? Okay, so they have given us the option 1 bone marrow, 2 lymph, and 3 lungs. So we know that white blood cells are made in the bone marrow, right? So the white blood cells will be found in the bone marrow. The second is lymph. We know that in the lymphs, in the lymphatic system, we find the lymphocytes. Lymphocytes are also a type of the white blood cells, so we can find the lymphocytes या फिर white blood cells in the lymph as well. Statement 3 over here is saying the lungs. Yes, we know that white blood cells are also found in the lungs. They are found in the alveolus of the lungs. Why they are found in the alveolus of the lungs? They are found in the alveolus of the lungs for the defensive purposes. So hence the answer over here is option A. 1, 2 and 3 all are correct. Let's have a look at question number 4 over here. It says that which row correctly describes the B lymphocyte. If I talk about the lymphocytes over here, we know that the lymphocytes are formed in the bone marrow. Then there comes the T lymphocytes, and then there comes the B lymphocytes. We know that the B lymphocytes are all formed in the bone marrow as well as they mature in the bone marrow as well. And if I talk about the T lymphocytes, so they are made in the bone marrow but they are matured in the thymus. So over here they are saying that they are processed in thymus, but processed over here you must know that the process means that they are made in the thymus. So this is incorrect statement that they are not made in the thymus, they get matured in the thymus. So yeah, and then they are saying that they release antibodies immediately after the formation. No, they are not going to release the antibodies immediately after the formation. What they are going to do is that whenever they are going to encounter antigen, yeah, so whenever the antigen comes and binds with the B lymphocytes, only then they are going to release the antibodies. So let me write the reason over here. When बैक्टीरिया एंटीजन या फिर कोई भी पैथोजन का एंटीजन comes and it binds with the B lymphocyte, then only it is going to release the antibodies. So hence our answer for this question is option D. Let's move on to question number 5 now and see that what they are asking to us over here. The immune response to a vaccination against a virus can be assessed by making three measurements. The statement 1 is saying that the concentration of the antivirus antibody in the blood. Statement 2 is saying the time taken to increase the antibody concentration significantly after the booster vaccination. And then the statement 3 is saying the number of virus specific lymphocytes per centimeter cube blood. They are then saying that what describes the immunity of an individual when the values of 1, 2, and 3 are all low 10 years after the vaccination. If we talk about the vaccination over here, we know that the vaccination is a type of the artificial active immunity over here. Why? Because we know that in the vaccination, the antigens, yeah, we can say that the weakened antigens are injected into the body of the person, right? And the antigens do not enter the body naturally. So what happens that in the vaccination, as there is the artificial active immunity and the antigens are inserted या फिर injected into the body of the individual. So what happens that there is an immune response that occurs which produces the memory cells. So we over here know that as the memory cells are being produced, so that will be a long-term immunity. So let's eliminate the incorrect options and find the correct option for ourselves. We know that they are talking about after vaccination. So if they are talking about after vaccination, so we know that if they are talking about vaccination, then there will be the artificial active immunity. So the passive immunity will be incorrect, right? And as the immune response has occurred because of the vaccination, so there will be no absence of the memory cells as well, and as well as there will be no low immunity also. Low immunity due to the absence of the antibodies. No, this is also our incorrect statement as it is not making any sense with the information we discussed above. Then option A is saying that active immunity due to the presence of the memory cells. Yes, option A is our correct answer over here. Yes, A, the active immunity is being established due to the presence of the memory cells over here. Question number 6 over here says that Which are the specific immune responses? We know that if I talk about the immune responses, there is a non-specific immune response, and then there is a more specific, या फिर जो है we can say that the specific immune response. The non-specific immune response is also known as innate immune system, या फिर innate immunity. And the specific immune response, yeah, specific immunity is also known as adaptive immune system. If I talk about the white blood cells involved in the innate immune system, we know that the white blood cells that are involved in carrying out a non-specific immunity in our body are the phagocytes over here. And if I talk about the specific immune response and talk about the white blood cells that are involved in the specific immune response, so they are the lymphocytes over here. Lymphocytes are the white blood cells that are involved in the specific immune response. And we know that lymphocytes are divided into B lymphocytes and they are divided into T lymphocytes. The B lymphocytes then divide into the plasma cells as well as the memory cells. What do the plasma cells over here do is that the plasma cells secrete the antibodies. The best way to learn the chapter of the immunity is through the flow charts. And if you draw the flow chart and relate the things, the chapter of the immunity becomes very easy for you. So what they're asking to us in the question that which are the specific immune responses. So we know that the specific immune responses carries out the production of the antibodies. Why? Because in the specific immune response, the B lymphocytes are taking part and they are differentiating themselves into plasma cells that are secreting the antibodies. So statement 2 is correct. If I talk about the statement 1, which is the phagocytosis, we know that the phagocytes are a part of the innate immune system, या फिर they carry out a non-specific immune response. So hence the option 1, या फिर we can say that the statement 1 is not a specific immune response. Then in statement 3, they are saying that the production of the memory cells. Yes, this is also a correct statement. Why? Because we know that in the specific immune response which they are talking about, the B lymphocytes made are also differentiated into the memory cells over here. So hence our answer over here is option C, 2 and 3 only. Question number 7 over here is saying that why does the white blood cell count increases in the people with the leukemia? We know for this question we must know that what is the leukemia. Leukemia is basically the cancer of the white blood cells. We know that what happens in the cancer is that the cells divide uncontrollably, या फिर the cell divides abnormally, right? So we can over here say that more lymphocytes are produced in the bone marrow because they are talking us about the condition of the leukemia, and leukemia is the cancer of the white blood cell, and we know that in cancer there is the abnormal production या फिर more frequent division of the cells. So what will happen that there will be more lymphocytes which will be produced in the bone marrow. Hence the answer over here will be option B. Question number 8 over here is saying that which statement correctly describes the lymphocytes. Let's look at these statements one by one and try to figure out that what is the correct statement over here. Statement 1 is saying that each B lymphocyte has the ability to make several types of antibody molecules. No, this statement is not correct. Why? Because they are talking about the each B lymphocyte over here. If I talk about the each lymphocyte, या पे each B lymphocyte over here, I know that each B lymphocyte is more specific to only one type या फिर one specific antibody. Each B lymphocyte is going to make one specific antibody which is going to be complementary to the bacterial antigen that has activated that B lymphocyte. And as it is complementary to only one specific type of antigen, so it is going to kill a specific type of a bacterial cell. Right? So option 1, yeah, we can say that the statement 1 is incorrect statement. Then in statement 2, they are saying that some B lymphocytes and T lymphocytes become the memory cells. Yes, already I have discussed above as well that B lymphocytes convert into the plasma cells as well as the memory cells. And if I talk about the T lymphocytes also, the T lymphocytes also differentiate into the memory cells as well. So option, we can say that the statement 2 is the correct statement for us. Then in the statement 3, what they are saying that plasma cells secrete antibodies in the blood plasma. Yes, this is the correct statement because I told you guys earlier also in the lecture that the plasma cells are secreting the antibodies. So statement 3 is also the correct option. Then if I talk about the statement 4, it says that some T lymphocytes stimulate the macrophages to kill the infected cell. Yes, this is the correct statement because this may they are talking about some lymphocytes, some T lymphocytes specifically. So we have studied about the T helper cells initially in this lecture. What do the T helper cells do? That it release the chemical known as the cytokines, right? If you guys remember, so T helper cells stimulate T helper cell release the chemicals which are known as the cytokines. The basic purpose of the T helper cell to release the cytokines is that it stimulates the response of the B lymphocytes as well as the macrophages. So this is what the cytokines over here do. So option, we can say that statement 4 is also our correct option. So hence the answer over here is option D. 2, 3 and 4 statements are correct. Question number 9 over here says that which sequence of events occur during an immune response. So basically, before we start to attempt this question, we must have a knowledge of the primary immune response in the first place. So this is the lecture slide of the primary immune response. Let's have a look at the steps of the primary immune response first. So what is going to happen that whenever a bacterial antigen or a foreign antigen is going to enter the body, it is going to be recognized by the immune system, and what will happen that the bacterial antigen will bind to a specific clone of the B lymphocyte. The specific clone of the B lymphocyte means that clone is the group of cells, a group of identical cells. You can say that the clone is a group of identical cells, right? And what will happen that when the bacterial antigen will bind to a specific clone of B lymphocytes, it will activate that clone and the clonal selection will take place. अच्छा, अब specific clone का ये मतलब है that the bacterial antigen is basically complementary to a specific receptor at the B lymphocyte, right? तभी तो receptor antigen जो complex हमारे पास बनेगा, it would be because the bacterial antigen is basically complementary to the B lymphocyte. And this clone के साथ that antigen will be complementary, it will activate that clone, and this is known as the clonal selection. Then what is going to happen that the particular clone, that particular clone जो अभी activate हुआ है of the B lymphocyte, is then going to divide by mitosis and then they are going to increase in number, right? So this is basically termed as the clonal expansion. व्हाट विल हैपन दैट सबसे पहले हमारे पास क्या है, क्लोनल सिलेक्शन हुई। ठीक है, क्लोनल सिलेक्शन के बाद हमारे पास क्या हुआ कि द बी lymphocytes then start to divide by mitosis and increase in number, and then the clonal expansion takes place. And then after the clonal expansion, what will happen that there will be some B lymphocytes that will differentiate into number one plasma cells, and then there would be some B lymphocytes that would be differentiated into the memory cells over here. राइट, अब जो है प्लाज्मा सेल्स की अगर मैं बात करूं तो प्लाज्मा सेल्स आर दो सेल्स which are secreting the antibodies. They are the antibody secreting cells and they have been differentiated from the B lymphocytes. की पॉइंट है, याद रखना है इस चीज को। ठीक है, इफ आई टॉक अबाउट द मेमोरी सेल्स ओवर हियर, सो मेमोरी सेल्स का क्या रोल होता है हमारी बॉडी में? मेमोरी सेल्स का रोल होता है दैट दे सर्कुलेट इन द ब्लड एंड आर हेल्पफुल इन द सेकेंडरी इम्यून रिस्पॉन्स। वी नो कि सेकेंडरी इम्यून रिस्पॉन्स हमारे पास वो इम्यून रिस्पॉन्स होता है दैट इज मोर रैपिड। ठीक है, सो दे आर द मेमोरी सेल्स विच हेल्प्स टू मेक इट मोर रैपिड, so they are the basis of the immunological memory as well. So let's now solve this question and let's see that what are the sequence of events which occur during an immune response. The sequence of events that occur during the immune response, हमें पता है कि सबसे पहले हमारे पास जो है it would be the recognition of the non-self antigen. For example, the bacterial antigen or any antigen of a pathogen would be recognized, right? After the recognition, what will happen that the clonal selection takes place, right? And when after the recognition, when the clonal selection has taken place, then what is going to happen that there would be the clonal expansion. And what happens in a clonal expansion? Clonal expansion may these cells divide by mitosis. The B lymphocytes will divide by mitosis over here, right? So सबसे पहले हमारे पास 3 है, then हमारे पास जो है वो statement 2 है, and then when the clonal expansion happens and the B lymphocyte starts to divide by mitosis, we know that B lymphocytes are then further differentiated into the plasma cells as well as the memory cells. What are the memory cells going to do? Memory cells are the basis of the immunological memory, या फिर they are going to stay in the blood and then they are going to circulate in the blood. And in case that particular antigen, that same antigen attacks in the future, so it is going to release the antibodies more rapidly. Then what is the plasma cell over here going to do? Plasma cell over here is going to secrete the antibodies. So we know that there will be first the development of the plasma cells and then there would be the secretion of the antibodies from those plasma cells. So hence the answer over here is option B. Let's move on to question number 10 over here. Let's see that what they are asking us to do in question number 10. They are saying that the graph shows how the body reacts following the exposure of the antigen for the first and the second time, right? This is the graph showing the concentration of the antibody against the time. What is the direct cause of the increase in the antibody production after the second exposure to the antigen? If I interpret the graph over here, I know that there is a first exposure when the body will be exposed to the antigen for the first time. So what will happen that there will be a primary immune response which will be initiated by the body. And when the body is going to be exposed second time to that particular antigen, then there would be a secondary immune response. And we know that what is special about the secondary immune response that there are the memory cells already in the blood in the second immune response. If I start to talk about the primary immune response, we can see over here that when the primary immune response is starting, like when there is the first exposure of the foreign antigen in the body, what happens that it takes a lot of time before it reaches a certain concentration of the antibody, right? And why it is taking a lot of time in reaching a certain concentration of the antibody? Because there are no memory cells over here, right? Why? Because there are no memory cells. What do the memory cells basically over here are doing is that they quickly divide and differentiate into the plasma cells, and then that plasma cells secrete the antibodies. right? So now they are basically asking that what is the direct cause of the increase in the antibody production after the second exposure to the antigen. So we can over here directly say that because of the memory cells, what is happening that there is the rapid increase in the antibodies over here. So which memory cells are going to cause an increase in the antibody production? We know over here that there are the B memory cells. Why? Because B lymphocytes differentiate into the B memory cells, and only B lymphocytes are a type of the white blood cells which we have studied produce the antibody. T lymphocytes are not capable to produce the antibodies. Only B lymphocytes are there to produce the antibodies. Hence, the answer over here is option A. Acha, also we can over here discuss one more thing, that why is this concentration falling? What is basically making the concentration of the antibody to fall in the blood? This antibody concentration over here is falling because the antibodies over here are being broken down in the liver. After the primary immune response takes place, what happens is that the antibodies are basically broken down in the liver. That's why the concentration of the antibody over here are falling.